Hello, listeners. I'm really happy to be sitting down with Adrian Gabirel Torres for our Immagina podcast, where we learn more about the research scientists are doing in the ribosomal translational field. Adrian, could you please introduce yourself and the work that you're doing?
Hi, Elisa. Yeah, I'm a research associate at the Gene Translation Lab in the Institute of Research in Biomedicine in Barcelona. And I am interested in understanding the mechanisms by which tRNAs, or transfer RNAs, regulate cellular physiology. So essentially, tRNAs are known to play an important role in protein synthesis, but in recent years, they have also been found to regulate other biological processes. So I study how the cell modulates this balance between the canonical and non-canonical tRNA functions in physiological and pathological scenarios.
And with your research, what do you hope to accomplish? What would be the best outcome of the research that you're doing?
Right. So defects in tRNA and tRNA biology in general have been linked to a wide variety of human diseases. So we hope to achieve a fine understanding of the different cellular pathways the tRNA act upon, and learn how tRNAs regulate them, so that eventually we would be able to find novel therapeutic strategies to tackle complex pathologies such as cancer, neurological disorders, or metabolic diseases.
And how does translation analysis impact your research? What do you do with it?
Yeah, so studying translation is essential in my line of work. So tRNAs are adaptor molecules that read the genetic code imprinted on messenger RNAs and translate it into amino acids that will be incorporated into proteins. Therefore, if we want to know how tRNAs exert their function, we must be able to monitor carefully the process of translation. For example, defects in tRNAs can slow down translation and cause premature termination of the protein synthesis, or may induce the incorporation of wrong amino acids into proteins, leading to mutated proteins with impaired function or proteins that are toxic for the cell. So this and many other possible outcomes of tRNA-mediated defective translation can lead to diseases.
And could you describe briefly in the future how Immagina Biotech could help your science?
Yeah, so for example, the analysis of ribosome occupancy within transcripts by ribosome profiling, it's a very powerful approach to study translation at codon resolution, but it is experimentally very challenging. So Immagina Biotech has developed this ribose technology, which we are starting to apply now, that can effectively purify actively translating ribosomes for ribosome profiling experiments in a very much simpler manner. So we find that to be able to simplify the experimental handling of samples is particularly critical when studying, for example, human biopsies. And since we are using materials from patients, in this sense, we believe these kits from Immagina can help us get better results by using these precious samples, right?
Well, thank you, Adrian. It was really nice to talk to you and also understand more of the work that you're doing. And thank you for taking again the time to talk to us. I'm really excited to give light to your research.